Hello everyone, my name is Sridhar Pushparaja and I am an integrated master's PhD student in the Department of AI Convergence at Chennai University. Today, I will be presenting a paper titled Reproducible Polarization Test Methods and Fair Evaluation of Polarization Data by using interconversion factors in a single chamber cubic microbial fuel cell with a brush anode. This was published in the Journal of Cleaner Production in 2023 by Professor Soki P. Jung. The paper focuses on enhancing the evaluation of MFC performance through the development of standardized and reproducible testing methods, addressing key challenges in the field of microbial fuel cell research. This slide provides a structure of presentation which includes the following sections: microbial fuel cell, introduction, background, objectives, MFC system, materials and methods, results and discussion, and finally the conclusion. Here is the pictorial representation of a microbial fuel cell. This is a bioelectrochemical system that generates electricity from the metabolic activities of microorganisms. MFCs have the potential to address both energy and water crisis by converting organic waste into bioelectricity. This is the anode compartment and here is the cathode compartment. The organic material such as acetate is introduced into the anode chamber where it is broken down by exoelectrogens, the exogenic microbes that are capable of transferring electrons externally. And the electron released by the exoelectrogens are transferred to the anode, then travel through an external circuit, producing an electric current. This flow of electron powers the electric circuit. Then the protons generated at the anode travel across the proton exchange membrane to the cathode side. This separation is important for maintaining charge balance and facilitating the electrochemical reactions. At the cathode compartment, the oxygen acts as an electron acceptor, so the electron flow from the external circuit into the cathode where they combine with protons and oxygen to form water or hydrogen peroxide depending on the specific reaction. So, in summary, this system generates electricity as a result of the microbial metabolism of organic matter at the anode and the reduction of the oxygen at the cathode. The flow of electrons through the external circuit is what powers electrical devices, and the transport of protons ensures the completion of redox reactions. Microbial fuel cells generate bioelectricity from organic waste and addressing energy and water crisis. MFC research have grown but still lacks reproducible methods and consistent data evaluation. Polarization tests are key but often yield inconsistent results due to system and microbial variations. Challenges like power overshoot and high internal resistance complicate data accuracy. Standardized reproducible measurement methods are crucial for advancing MFC research and ensuring fair comparison of results across studies. So, the MFC research has been conducted enormously in the last 2 decades. Reproducible method and fair evaluation are still the missing components in MFC research. The electrochemical methods and MFC systems are very different in each study. Their measurement and analysis were inaccurate. The power overshoot undermines data quality. ERC, the external resistor change method, is widely used in MFC research to evaluate polarization curves. It involves selecting and switching between external resistors and waiting for the system to stabilize at each settings. The ERC method can be time consuming due to the need for steady state conditions. The ERC method also produces limited data points which can hinder the detailed analysis of MFC performance. The accurate optimization of internal and external resistance enhances the power density and current output. The long-term microbial diversity and biofilm formation monitoring is essential for stable operation. LSV, which is called as linear sweep voltammetry method, is a faster alternative to ERC. It does not require the selection of external resistors, and also there is no need to select retention and stabilization time, which allows for quicker tests with more data points. LSV is much faster than ERC, and it rarely affects anodic microbial community and electrochemistry. Also, LSV generates plenty of data points. The main objectives of this study is to establish reproducible condition using low internal resistance cell. Also, to establish reproducible electrochemical test methods and to establish fair evaluation method of polarization data and to provide baseline information of a single chamber MF. This slide describes a single chamber cubic MFC used in this study. It is also known as the standard cell. It has the features of low internal resistance and it utilizes a single chamber cubic MFC design. It is equipped with the graphite brush as the anode. It is developed by Dr. Logan's group at Penn State, and also it is one of the most widely tested systems. It has a large body of accumulated data. Its compact and simple structure make it easy to operate. It typically uses 10 to 20 millimolar of acetate in 50 millimolar of PBS or CBS as the substrate. This study utilized 4 identical MFCs with a 28 ml chamber volume. The system used an activated carbon brush air cathode and a heat-treated carbon fiber brush anode. The anodes were inoculated with wastewater sludge, and the substrate consisted of 50 millimolar phosphate buffer saline with 10 millimolar sodium acetate. The polarization tests, cyclic voltammetry, and electrochemical impedance spectroscopy were performed to evaluate the system's performance. There are two polycarbonate single-chamber MFCs with carbon fiber brush anodes, and the anode is heated to 450 degrees centigrade for 30 minutes before biofilm inoculation, and the cathode is a stainless steel mesh with activated carbon, carbon black, and PVDF assembled with titanium, and anodes inoculated with wastewater operated at 1000 ohm in 50 millimolar PBS for 9 days. The MFCs run at 100 ohm for 9 weeks before electrochemical tests. LSV, EIS, and CV performed using a potentiostat. 100 ohm matched internal resistance for optimal power. The polarization curves were generated using ERC and LSV at a scan rate of 0.1, 1, and 10 MB per second. Cyclic voltammetry measurement current and voltammogram areas to assess anode and cathode activity. Impedance analysis showed charge transfer dominated total cell resistance. COD was measured after 9 to 18 hours by filtering samples and using a COD kit. Coulombic and energy efficiencies were calculated from current, voltage, and COD reduction. Hence, power overshoot is a major problem. It is necessary to eliminate the power overshoot. And here is the way to eliminate the power overshoot. To eliminate power overshoot, the optimal resistance operation time (ORT) and open circuit time (OCT) were determined. Before electrochemical tests, MFCs were run at 100 ohm for 3 hours in a fresh medium with 10 millimolar acetate. For polarization tests, the circuit was disconnected for 90 minutes, then reconnected to a potentiostat for 30 minutes in an open circuit state. Here is the detailed results of the polarization tests. When using the faster line sweep voltammetry method, the scan rate of 10 MV per second, the maximum power density achieved was around 835 millivolt per meter square. Here, the maximum power density, optimum current density, optimum external resistance were determined. Power increases with faster scan rates, which is LSV10, 1 minute shows the highest, and ERC 240 minutes shows the lowest. The faster scan rates, like LSV10, reduce polarization resistance. Also, the faster scan rates lead to higher current and lower resistance, boosting performance. The measurement time is decreasing in the order of ERC, LSV 0.1, LSV 1, and LSV 10. As the polarization measurement time decreased, both power and current densities increased. Faster measurements led to lower polarization resistance and increased power production. This highlights the benefits of faster polarization methods for improving the reproducibility and performance of microbial fuel cells. The RST values measure the reproducibility of the data, with lower values indicating higher consistency in performance measurement. As the measurement time for polarization decreases, the RST values also decrease, showing improved reproducibility of results. Faster polarization tests provide more reliable data, as demonstrated by the lower RST values for key performance parameters. Considering measurement time and data reproducibility, we recommend LSV1 and LSV 0.1 as the optional measurement methods in the single chamber cubic MFC experiments. Here, the S-shaped display is the anode, it indicating efficient charge storage and release. And also in the cathode, it shows an almond shape reflecting distinct electrochemical behavior compared to the anode. The impedance measurements for the anode and cathode were taken at their optimum electrode potentials. Around 70% of the total impedance was from the cathode, while the anode contributed approximately 30%. The primary component of impedance was charge transfer impedance. Lowering the charge transfer impedance is crucial for enhancing electrode performance. Diffusion impedance was observed only in the cathode, accounting for 4.8%. When it comes to the interconversion factors, for fair comparison of different polarization data in the single chamber cubic MFC systems, the interconversion factors were calculated. For a more stringent comparison, medium composition, solution volume, operation method, reactor structure should be considered. The values calculated by the interconversion factor are estimates. They can be slightly different from measured values. In conclusion, a reproducible measurement method and fair data evaluation were established for microbial fuel cells. Optimal resistance operation for 3 hours and open circuit time for 2 hours effectively prevented power overshoot. Faster polarization tests increase reproducibility as indicated by lower relative standard deviations in cell performance. Anodic polarization resistance was lower than cathodic polarization resistance, contributing to better overall performance. Impedance and capacitance tests revealed that charge transfer impedance dominated, especially in the cathode. This study provides insights to improving experimental accuracy and reproducibility in the MFC systems. Thank you.